Hi, good morning. This is a neuro class, the control of ocular movements. In the last class, we saw that the sensation of vision is one of the most important sensory modalities as compared to the sensations from the skin or gustatory or visual sensations. And therefore, the optic nerve has about 1,000,000 nerve fibers. I told you how it pierces the sclera at this lamina cribrosa sclera, which is one of the weakest parts of the sclera, and you can see these 1,000,000 fibers of the optic nerve which become myelinated once they exit from the retina. And again, this is just to show you, this is the million fibers of the optic nerve, and how the dura continues as the sclera, the pia arachnoid continues as the choroid, and the optic stalk and the optic cup, then they differentiate into the retina. You've already done the optic pathway. And I mean, just a brief recapitulation because we are waiting for all the students to come back. You saw that from the diencephalon floor there develops this optic cup. This is going to differentiate, and this forms the stratum pigmentosum. This is the stratum nervosum. There's a visual part of the retina which stops short at the ora serrata, and then it just continues as the double layer of epithelium beyond the ora serrata, which covers the inner aspect of the ciliary body and the iris. This is from the previous class. We also talked about the anterior pole, the center point of the corneal curvature, the posterior pole, and there was a visual axis which is very important for today's class, which is from the anterior pole to the fovea. You also know, when I talked about the layers of the eyeball, I told you that the retina, I talked about all the layers, and I told you that there are certain specializations in the retina. And the most important, there was a blind spot where there are no rods and cones, so it's insensitive to light. That's known as the blind spot, and this was the area which is the area for most acute vision. This is the fovea center. I talked about the macula, and in its center you have the fovea, and the depression is known as the foveola. Now the most important thing to remember about this is that this fovea in this region just has very slender cones, and here also you find that only the cone pedicels are present, and the cone bodies are also displaced laterally, right? And this part gets nourishment from the lamina of choriocapillaries, which is best developed in this region of the fovea. So there is no capillaries found in this region, and the blood supply from the central retinal artery is not supplying this region. It is supplied by nutrition from the lamina of choriocapillaries. So this is the area for most acute vision, and this area is the last to mature at about 4 months after birth. And so you can see that it is the area where you have slender cone pedicels, and the cone bodies are also displaced laterally. So you find this part of the retina is the thickest, the parafoveal region. So you've already discussed about the optic pathway. Just a brief recap. So you have the optic nerve, the optic chiasma, where the nasal fibers decussate. You have the optic tract, which divides into two: a medial part which goes as through the forms the superior brachium, which ends in the superior colliculus and the pretectal hoop of nuclei. From the lateral geniculate body, this lateral part, it enters the lateral geniculate body where you have the third order neurons of this pathway, the first being the bipolar neurons, the second the ganglion cells, and here you have the third order neurons, and you can see this is these fibers are the fibers of the geniculo-calcarein tract, which end in the visual cortex, which is also known as the striate cortex because of the presence of stria or the band of Ballarger or the line of Gennari, right? So you have the visual cortex, which is a sensory cortex, okay, which is found on the walls and the lips of the calcarein sulcus. You know where is the calcarein sulcus? You trace it from the medial aspect, okay, and then you have this is an axial sulcus, the calcarein sulcus, and it's on its walls and the lips you have the the primary visual area, which is area 17, and then you have the area 18 and 19, which is the para and the peristriate areas. Okay, so for visual reflexes you have a different pathway, and we'll just discuss that briefly. Okay, so you already know that when you shine eye in one light, the pupil of that eye constricts. Okay, there's pupillary constriction. The pupil of the opposite eye also constrict, which is known as the consensual light reflex, and that happens because you find that there's decussation of fibers of the optic in the optic chiasma, and also you find that fibers from the pretectal nuclei, they go to the Edinger-Westphal nucleus of both the sides. Okay, so you have the direct and the indirect light reflex, and then the fibers are going to pass these general visceral efferent fibers, they pass along on the superficial surface of the oculomotor nerve, and relay in the ciliary ganglion, from which the short ciliary nerves are then going to supply the ciliary muscle as well as the sphincter pupillae. For the light reflex, you require the sphincter pupillae, right? So today the lecture is on control of ocular movements. So we will just discuss the most important cranial nerve nuclei which are involved and the other centers which are involved in this pathway. So you've already discussed about the third, fourth and the sixth cranial nerves. Dr. Basure had done that in his lecture, but I'll just briefly recap that. I would just like to tell you that the cranial nerve nuclei which are concerned with ocular movements are also known as the ocular motor nerve nuclei, and these are the third, fourth and the sixth cranial nerve nuclei. Now all these cranial nerve nuclei, they have type A and type B neurons. The type A neurons are the general somatic efferent neurons, the general somatic efferent neurons which are going to innervate the neurons, or which are going to innervate the muscles. And these muscles, one neuron is going to innervate about 6 muscle fibers, and these are the somatic, general somatic efferent neurons. And then you have type B neurons, which are the internuclear neurons. Now these are very, very important, because as we see that when the eyes move, both the eyes show conjugate movements or yoke movements, which are movements in which you have both the eyes moving together, okay, in the same direction through the same amount. And this function is subserved by the type B neurons, which are the internuclear neurons. Okay, so type A are the general somatic efferent neurons, and you have the type B neurons. Okay, now let's just see the third nerve nucleus. You know that the third nerve nucleus lies in the midbrain, in the periaqueductal gray, at the level of the superior colliculus. It lies on the ventral aspect. You have these cranial nerve, this is a third nerve, and here in this third nerve nucleus you have various subnuclei which innervate different muscles that the third nerve supplies. For example, it supplies the superior rectus, the inferior rectus, the inferior oblique and the medial rectus. So each of these there is a separate subnuclei. Okay, it's also important to know that the fibers of one side supply the same side muscles, except the superior rectus is supplied by the oculomotor nerve of the opposite side. So this nerve passes ventrally and emerges on the medial side of the this interpeduncular fossa, or on the medial side of the cerebral peduncle. You know that half the midbrain is known as the cerebral peduncle. This is the crus cerebri, and this is known as the tegmentum. And dorsal to the aqueduct, this part is known as the tectum. So the third nerve emerges on the medial side of the cerebral peduncle. Okay, in addition to that, as I told you, in addition to this, now at the rostral levels, there is another nucleus which is very important in the third nerve, and that is the Edinger-Westphal nucleus, which has the general visceral efferent neurons. The general visceral efferent neurons, which are going to give rise to the peripheral parasympathetic fibers, which run on the these fibers are very superficially oriented. These are very superficial in the third nerve. So if there is compression from any exterior source, these are the fibers which first get compressed. Okay, and at the more caudal levels, you have another midline nucleus which I had discussed about in my previous class. That was the nucleus of the levator palpebrae superioris, and it's a single midline nucleus, and its axons diverge and go to both the sides, this one to the third cranial nerve nuclei. In addition to that, in the third nerve you find that there are some accessory oculomotor nerve nuclei which are found in the reticular formation. Now these nerve nuclei are important for the class today, not all of them but some of them, and I'm just going to name them. First is the rostral interstitial nucleus of the medial longitudinal fasciculus, the rostral interstitial nucleus of the medial longitudinal fasciculus, the interstitial nucleus of Cajal, the nucleus of the posterior commissure, as well as the nucleus of Darkschwitz. These are accessory oculomotor nerve nuclei. Accessory oculomotor nerve nuclei. Okay, and these nerve nuclei have a very, very important role for execution of these conjugate eye movements, and we'll just discuss about them in more detail. Then you have the fourth nerve nucleus. Now the fourth nerve nucleus, you find it's lying at the level of the inferior colliculus. It lies again in the periaqueductal gray, and you see that the fibers emerge, but instead of passing ventrally like all the cranial nerve nuclei come out, these fibers pass dorsally and they decussate. And as you know, the trochlear nerve is given the name trochlear because the muscle it supplies, that is the superior oblique, passes through the trochlea, that is its functional origin. Okay, so this is known as the trochlear nerve, and you can see that the fibers of the trochlear nerve, they decussate. So each trochlear nerve, for example, of the right side is going to supply the muscle of the left side. So similarly here, the superior rectus is supplied by the subnuclei, which is on the opposite side. Okay, so you find that the fourth nerve emerges on the dorsal aspect, the fibers decussate here and then emerge out on the lateral side of the cerebral peduncle between two arteries. You should know which are those two arteries. And then we'll do the sixth nerve nucleus. You've seen the sixth nerve nucleus, it lies below this is the open part of the pons. I mean, this is the part of the pons, and this is the floor of the fourth ventricle. And you can see this part here, this elevation is known as the facial colliculus, which is caused due to the internal genu of the facial nerve. And below that you have this nucleus, which is the abducens nucleus. And we can see that the fibers of this sixth nerve, they emerge out on the ventral aspect at the pontopyramidal junction. Right? So again, I just wanted to show you here that apart from the main, this at the level of the superior colliculus, you have the third nerve. It has also the general visceral efferent, the nucleus, which is the Edinger-Westphal nucleus, which is lying at the more rostral levels. You have the levator palpebrae superioris nucleus, which is at the more dorsal caudal levels. But in addition to that, you have some very important accessory oculomotor nerve nuclei, which you can see very clearly here. This is the rostral interstitial nucleus of the medial longitudinal fasciculus, nucleus of Darkschwitz, the interstitial nucleus of Cahal, and the nucleus of the posterior commissure, which is important for our class today. Okay. Now I'll just tell you some important areas which are concerned with vision. Now it's important to know that this ocular movements are a part, in mammals, are a part of cognitive behavior, right? So they are under voluntary control. Some are involuntary, but they are majorly voluntary control. And since they are under voluntary control, we are able to decide what we want to see, when we want to see, and whether when we see any, for example, you are looking here at the slide, you're looking at me, there must be so many things in front of you, maybe like a clock or whatever. And it's your brain which actually, the cerebral cortex, which actually decides what is important for you, what demands your attention. So you selectively filter the information or the stimuli which are there in front of you to choose the one which is of most important to you, right? So that is known as visual attention, which is selectively filtering the visual information which is in front of you. And there are different areas of the cerebral cortex which are involved in these functions, and I'll just talk about them. The most important is, as I told you, there was a calacrine sulcus which is found in the lips and the walls of the calacrine sulcus, that is the visual cortex. Okay, and then you have this area here, where you have the para and the peristriate areas, area 18 and the 19, which are also known as the secondary visual areas. So the image that is formed in the visual cortex actually lacks detail. It is not analyzed. It's just a 2D image now has to be converted into a 3D image. Okay. Then you have certain areas here. Now when you read your books, there is the term has been mentioned, the middle temporal and the medial superior temporal areas. Now when I talk today of these two areas, the area that I'm talking about is this junctional area between the anterior part of the occipital cortex, the posterior part of the parietal cortex, and this portion here of the temporal cortex. Okay, that is known as the middle temporal or the medial superior temporal areas. These are the names which have been coined in monkeys, right? In addition to that, there is area 7, which is the area in the lateral parietal area, this area here, which is concerned with visual attention. Now this is the area which actually determines that out of so many stimuli things which you are seeing in the visual field, which is the one which is important for you, to which you need to look. And the main aim of all these oculomotor movements is this, that whichever thing you think is important for you, you try to focus your eyeball to that so that the image of the object falls on the fovea. So the most important, the main aim of all these ocular movements is that the image of the object falls on the fovea. Okay, so first aim is to focus your eyeball in such a way that the image of the object falls on the fovea, and the second thing is that it remains on the fovea till you are focused on that part. And so the center which actually is determining is visual attention. Then, as I told you, that all these functions are part of cognitive behavior. That means you decide what you want to look at or what you don't want to look at, and when you want to make, for example, the 7 decides it's important, and so then these fibers relate to area 8, which is the frontal eye field. The frontal eye field is found in the middle frontal gyrus. It's area 8, and it's very important area because that's the center for conjugate movements. Okay, and now this center, that is the frontal eye field, is the one, once it gets information from area 7 here to these association fibers to the area 8, this is the one which is then going to give command for executing the movements. Okay, and then you have higher visual association areas, which are area 39 and 40. Now when you're executing these visual movements, you also have very important role of both the as I told you, of the cerebrum as well as of the cerebellum, especially the part of the the vermal, the inferior vermis, and the floccular nodular lobe. Then you also have the reason is that these are the ones which are going to actually calculate how much the eye has to move, how much the head has to move, especially when you're following some object which is moving across your visual field, to actually calculate how fast that object is moving or how much the eye has to move, or so that you do not move past that object, so the stability is maintained, the tone of the muscle. This is all done by the cerebellum. Then you have the vestibular nuclei, which are going to receive the information from the maculae of the utricle and the saccule and the semicircular canals. You all know that the vestibular nuclei are situated in the floor of the fourth ventricle in the lateral part. There are 4 medial, lateral, superior and inferior. But the most important, and you've already done the vestibulospinal tract, which ends at different levels of the spinal cord and is important for tone and posture and equilibrium of the postural anti-gravity muscles and muscles of the limbs. And you have the medial vestibular nuclei, which is very important because that is the one which is going to end at the cervical levels of the spinal cord, and this is required for the coordinated movement of the head and the eyes. So movement of the neck, so this is a very important pathway. And some fibers of these, and the medial vestibular nuclear fibers, they pass in the medial longitudinal fasciculus. Now what is the medial longitudinal fasciculus? It's a very important midline tract. It's heavily myelinated, and this tract consists of fibers from the medial vestibular nucleus, some fibers also from the superior, the medial vestibular nucleus. And this medial longitudinal fasciculus is very important because this interconnects the various cranial nerve nuclei. For example, you know that if suppose I have to look right, my right eye, the eyeball will be abducted towards the right side, and the left eyeball would be adducted towards the left side. So for the coordinated movement of both these eyes, so the medial rectus would contract of this side and the lateral rectus of this side. So the coordinated movements of the three cranial nerves, you have the connections is through the medial longitudinal fasciculus. This is a heavily myelinated tract, and because of its length, that's responsible for its vulnerability, and any disorders, demyelinating disorders, can affect the medial longitudinal fasciculus, resulting in internuclear ophthalmoplegia. And this actually happens in multiple sclerosis. Okay. Then you have the superior colliculus. The superior colliculus, as you all know, it's the center for visual reflexes. It's also a very important visuomotor integrating center, because as I'll talk to you today, that it's a very important center today, because for this lecture today, because this center is again a multi-layered structure. It has 7 lamellae. It has neurons which respond to visual stimuli, which are in the first 3 layers, and it has neuron in the deeper layers, lamina 4 to 7, which responds to visual as well as somatic, visual, somatic and acoustic stimuli. So this is the one. So not only it's a center for visual reflexes, it's also a relay center, and it's a center which receives information from the visual, somatic and the acoustic stimuli. It gets information from, for example, the inferior colliculus. It gets information from the spinal cord, like the spinotectal tract. It gets information from the cerebral cortex, from the frontal eye field. Okay. And so the superior colliculus is a very important center, and also the rostral part of the superior colliculus has a very important zone which is known as the fixation zone, and I'll just emphasize what is the importance of fixation. Okay, now why I got this slide was just to emphasize to you that when you have many things in your visual field, it's actually you decide what you want to look at. For example, there are so many things on this slide, And so it's now your area 7 is activated, and that's the center which is for visual attention. So the selective filtering of visual information is known as visual attention. So when there are so many things in front of you, if you're looking at this slide or this slide or this slide, this picture, then this picture is most sharply focused and these are out of focus. You must be feeling the same thing. So it's very, very important. So it's your brain which decides at a particular moment what is the stimulus which is most important for you, and then you move your eye to that particular area and you focus your fovea on that particular image, right? So that is known as visual attention. Then there are two pathways. As I told you that the image which is formed in the visual cortex is a two-dimensional image, but there are so many parts of an image. For example, you view the world as a whole. For example, if you are thinking of a tennis ball or a cricket ball, you all know when you think of a tennis ball, you think of a soft green ball which it won't hurt you. You think of its size, you know that it is made of rubber, and for example, you think of a cricket ball, you think of a red ball, cork ball, hard ball, and it has those cotton thing that around the center. So all these are attributes of any image. Now when you think of an image, you think of its position, its velocity, its thickness, its depth, and where the object is. Now that is deciphered in the dorsal pathway. This is the dorsal or the parietal pathway. So once the fibers have relayed from the bipolar, the ganglion, to the lateral geniculate nucleus, the geniculocalectrine fibers, the fibers pass from the area 17 to areas 18 and 19 and then to the posterior part of the parietal cortex by the dorsal parietal pathway. And this dorsal parietal pathway, the main function is to calculate the motion, the depth, the velocity of the object, or where the object is. So where the object is calculated by the dorsal parietal pathway, right? And the attributes of color, contour, shape, contrast, these are then deciphered by the ventral pathway which goes to the inferior temporal cortex. Okay, so that is the ventral temporal pathway. Okay, so if you want to calculate the position, the velocity, the depth, the distance, or where the object is, the pathway that is involved is the dorsal parietal pathway. If you want to see the shape, decipher based on the shape, size, contour, color, it's the ventral pathway which is relaying to the inferior temporal cortex. Okay, now we'll do the different types of movements that the eye has. Firstly, what is gaze? Gaze is nothing but the coordinated movement of the head and the eyes. It's the coordinated movement of the head and the eyes. Okay, and the main function of gaze is to stabilize the eyeball in the orbit or to stabilize the orbit in space. Okay, to stabilize the eyeball in the orbit or to stabilize the orbit in space. Okay, and the main aim of all these movements is, as I told you, that once you select something which is important for you to look at, which demands your visual attention, you focus your fovea on that. The eye moves in such a way so that the image of the object falls on the fovea and remains there. Okay, so there are two types of movement, basically the conjugate movement and the disconjugate movement or the disjunctive movements. So the conjugate movements are those movements in which the eyeballs move in the same direction simultaneously through the same amount. So they move simultaneously in the same direction in the same amount. And then there are disjunctive movements in which so the conjugate movements are known as yoke movements, eyeballs moving same direction, same amount. And there are disjunctive movements, which are movements in which the eyeballs are moving in opposite direction and maybe to different amount. Okay, now we'll just discuss what are the different types of conjugate movements. The conjugate movements are classified into saccades, smooth pursuit movements, optokinetic movements, vestibular ocular movements, and fixation. Okay, and disjunctive movements are vergence. Okay. Okay, now we'll do what are saccades. Okay, now the saccades, our saccades can be of three types. They could be vertical, horizontal, but this would be a vertical saccade, this would be a vertical saccade, this would be a horizontal saccade, and this would be an oblique saccade. An oblique saccade would involve both vertical and horizontal saccades. Okay, so all these movements, be it a saccade, smooth pursuit, optokinetic, vestibular, ocular, or fixation, are all conjugate eye movements. Okay, Now let's do what are saccades. Now whenever you read anything or whenever you look at anything, for example here it's shown you, you execute what are known as saccades. Whenever there is a stationary object, you look at an object, you do not execute a smooth movements, your eyes execute very rapid jerky movements. They execute little jerky movements followed by stoppage points or pause points. For example, you can see here that when the person is looking at this woman's eyes, this woman's face, sorry, the first he looks at the eyes and the person is executing rapid jerky movements, right? So you can see these movements, and these are followed by these stoppage points. So these little jerky movements followed by these stoppage points constitute what are known as saccades. Saccades, 90% saccades are voluntary. Okay, and the centers are in the frontal eye field, because these are by command from the frontal eye field area 8, and if they are involuntary, which are very few, these are by the superior colliculus. Now so what are saccades? These are when you are looking at a stationary object, or when you are reading the newspaper or reading your book, you do not see it in a smooth fashion. Your eyes execute rapid jerky movements followed by stoppage points. Now these rapid jerky movements followed by stoppage points, these stoppage points are known as the fixation points. Okay, there are different set of neurons which are active here to cause these rapid jerky movements, and when the eyeball stops here, these are the pause points, the fixation points. There are different neurons which are active, and these are equally important because they help the fovea to focus at a particular point so that the eye does not drift back. Okay, so that the eye does not drift back. These are very, very rapid movements. The eyeball moves at a rate of about 900 degrees per second, and these saccades not only are made in response to visual stimuli, they can also be made in response to the acoustic and the tactile stimuli. Okay, so the center for these conjugate saccade, these movements are the frontal eye field. Now I told you that these saccades could be horizontal, vertical or oblique, and oblique require both the horizontal and the vertical to be active, and I'm just going to explain these pathways in detail. They have been given beautifully in bar, but I would be explaining them in a little more detail for you to understand, and it's important for all the postgraduates to understand the detailed pathway, but for the undergraduates it's just enough to read bar. Okay, now for the horizontal saccades, the center where there are, okay, now I'll just okay, okay, just remember that for horizontal saccades, the center which initiates the movements is the paramedian pontine reticular formation. The paramedian pontine reticular formation is also found in the pons just near the abducent nerve nucleus. Initially they were constituting what is known as the para-abducens complex, but since they are not a distinct clinical entity, they are now coined separately. That is, now it is known as the paramedian pontine reticular formation. That is the center where you have the neurons which are going to issue command to the sixth nerve nucleus to immediately start firing. So these are the ones which are going to be having the neurons, the burst neurons, okay, which I'll just talk to you what they are. I'll just explain to you that. Then you have the nucleus prepositus hypoglossi. That is the ones which are going to cause the eyeball to stop. So you have here the neurons which are going to be the tonic neurons which are going to cause fixation. So just remember, for horizontal saccades, the neurons which are going to be sending these information to the sixth nerve nucleus is going to be the paramedian pontine reticular formation, and then you have the nucleus prepositus hypoglossi which has the tonic neuron where the eyeball actually stops. Okay, then you have the vertical saccades. For the vertical saccades, you have nuclei which are the accessory oculomotor nerve nuclei found in the mesencephalic reticular formation. That is the rostral interstitial nucleus of the medial longitudinal fasciculus, which is going to again be having the burst neurons, and so that which are going to initiate these movements, and to stop these movements for fixation, you have the tonic neurons which are situated in the interstitial nucleus of Cajal. Again, I am repeating, when you have to look at a stationary object, like you are looking at somebody or you are reading something, you execute movements which are known as saccades. Saccades are voluntary movements, and these are movements which are very, very rapid jerky movements which are followed by stoppage point or fixation points. See, these are rapid jerky movements followed by these stoppage points. Okay, These are very rapid, so they occur at a rate of about 900 degrees per second. These are usually voluntary movements. Then you have movements which are smooth pursuit movements. Now I don't know, the video is not working, but what I wanted to show you was, suppose a bird moves across a visual field and you follow that bird. Those movements are known as smooth pursuit movements. Now the eyes follows the bird, but you have to have information from the cerebellum, from the vestibular nuclei, to actually calculate the velocity with which your eye moves so that it matches with the velocity of the bird moving across the visual field. Okay, so these are known as smooth pursuit movements, and what's important is smooth pursuit movements are involuntary movement. Involuntary movement, you are getting information from the cerebellum, right, and from the vestibular nuclei, and these are then giving command to your eyes to actually execute smooth pursuit movements. So these are involuntary movements. These are much slower. They're about 100 degrees per second. And in addition to that, what's most important is that these smooth pursuit movements cannot be made in response to acoustic stimuli or tactile stimuli, because those would be a jerky, rapid movement. You cannot do a smooth pursuit movement in response to an acoustic or a tactile stimuli. So these are smooth pursuit movements. Actually, I wanted to show you this bird flying around to make this nest, but the video is not working. But what I wanted you to see was that these are involuntary movements. They are slower movements. They track a moving object in the visual field, and they cannot be made in response to an acoustic or auditory stimuli. And the center, which is for the smooth pursuit movement, just like you had the frontal eye field there, the center for the smooth pursuit movements is the junctional area between the occipital, temporal and the parietal cortex. Now this area in the books has been named as the middle temporal and the medial superior temporal area, but it's nothing but it's the anterior part of the occipital, posterior part of the parietal cortex, and the junctional area with the temporal cortex, which is the area for smooth pursuit movements. Okay, so we've seen what is a saccade, we've seen what is a smooth pursuit movements, and then you have optokinetic movements and the vestibulo-ocular movements. We've seen what is fixation. Fixation is an active process which keeps the eye fixed. The eyeball does not move during that, so the tonic neurons are active. Then you have the vestibulo-ocular movements. Now what are vestibulo-ocular movements? Vestibulo-ocular movements are those movements in which, okay, so just 1 minute. These movements, the saccades, the smooth pursuit movements and the fixation, are movements of the eyeball in the orbit. Okay, there is no movement of the head. In optokinetic and vestibulo-ocular movements, the head also moves. So the head moves and the eyes move, right? Now vestibulo-ocular movements are those movements in which there is just a rapid movement of it is a brief movement, a brief rapid movement. So what happens when you have to execute just a small movement, because the eyeball has less inertia as compared to the head. So what happens, the eyeball moves first and then the head moves and comes to lie in its new position, and then once the head has moved and comes to lie in a different position, the eyeball then moves in the opposite direction to come to lie in the center of its new position. Are you getting why? Firstly, the eyeball moves to look at a particular object, right? These vestibulo-ocular movements get input from the vestibular system. Now vestibulo-ocular movements occur, these are very brief movements, brief rapid movements. So what happens when you execute a vestibulo-ocular movement, since the head has a much larger inertia because of its mass as compared to the eyeball, the eye moves first. Okay, so the object now focuses on the fovea. Then the eyeball moves, then the head moves, so the eyeball has moved, it has focused on the fovea, the image. Then the head begins to move, and once the head begins to move and reaches the final position, that means the eyeball has also moved, so the eyeball moves in the opposite direction and comes to lie in the center of its new position. So the eyeball moves first, then the head moves, and then the eyeball moves in an opposite direction to come to lie in the center of its new position, again so that the object focuses on the fovea. So this is the same thing that you see in the doll. When you move the doll's head, roll the doll's head up, the eyeballs roll down. Then you have the optokinetic movement. The optokinetic movement are slow sustained movements. They are sustained movements of the head and eye. When you have a sustained movement of the head and the head and the eye move together. Okay, so there is no vestibular ocular reflex that is suppressed during that time. So in optokinetic movements, the head and the eye move together, okay, to look at a particular object, right? So this is about the conjugate movements of the eye. Then you have the disjunctive movements. Disjunctive movements are movements when you are looking at distant and near objects. You must have seen that when you are looking at a distant object and then you look at a near object, the distant objects appear blurred, and when you focus on a distant object, the near objects appear blurred. Okay, and only when you are looking at objects more than 100 feet, it appears that you are looking through one eye, because the image formed on both the retinas is nearly identical. But what's important is, what is the stimulus for disjunctive or the Vergence movement? It is blurring. There is disparity. For example, both the eyes are about 6 centimeters away, at a distance of 6 centimeters. The horizontal distance between the two eyes is about 6 centimeters. So, for example, if I'm looking at this whiteboard, it's nearer to my left eye as compared to my right eye. Okay, so the images formed in both the retinas are slightly different, right? So the eyeballs will, to look at a near object, the eyeballs will converge, and to look at a distant object, the eyeballs will diverge, and they might diverge through different amounts, and the convergence could also be through different amounts. Okay, so disjunctive movements are those movements in which the eyeballs move in opposite direction and may be through different amounts. Okay, now you have three types of movements. Now let's take the example of a saccade. I told you that the saccades are rapid jerky movements followed by stoppage points. Now you've seen that you have 100 things going on in front of you. Maybe, for example, I'll give you this example. I am speaking. You're now listening to me. Even if you have 5 or 6 friends who are sitting with you, and even if they are talking, you really don't pay attention to that. I hope so. So that is because your ears have decided that this is important to hear. Similarly, when you get a command from the parietal cortex, the posterior part of the lateral intraparietal area 7, it says that this structure is important for your visual behavior. Okay, this sends a command to the frontal eye field, and at that time you have the firing of neurons, because you decide what is important for your visual behavior. You're looking at 100 things. It's not that you look at all the 100 things. Your eyeballs don't keep moving all the time, right? They remain stationary. You only move an eyeball when you think something is important for you. And there are neurons which are preventing your eyeball from moving all the time. Okay, and those neurons which prevent your eyeball to making movements which are not required are the omnipause neurons. And when a movement has to be initiated, you have neurons which are the burst neurons. Okay, these are neurons which fire at a very rapid rate, about 1000 hertz, and so they are able to stimulate the neurons in a particular nucleus so that they can send information. They can stimulate the muscle to contract. Okay, in addition to, so the burst neurons are the ones which are firing very rapidly only when a movement has to be initiated. Okay, those are known as the burst neurons. When a movement has to be initiated, they fire very rapidly. Now, as I told you, for example, if my right eye has to look on the right, the lateral rectus of the right side contracts and the medial rectus of the opposite side. So the lateral rectus of the opposite side has to get passively lengthened, and for that you have the inhibitory burst neurons. Okay, the inhibitory burst neurons. Okay, so burst neurons are the ones which are going to initiate the movement. Inhibitory neurons will allow for the passive lengthening of the opposite side muscle, and the tonic neurons are the ones which are going to help in fixation. As I told you, for the horizontal saccades, the fixation is by the nucleus prepositus hypoglossi. For vertical saccades, the nucleus is interstitial nucleus of Cajal, and I also told you that these tonic neurons are also situated in the rostral part of the superior colliculus. Then, to prevent the eyeball from making unnecessary movements, you have neurons which are known as the omnipause neurons. Omnipause neurons. These are neurons which are active all the time and are only suppressed when you get a command to make a eye movement. Okay, so omnipause neurons fire all the time. They are active all the time, and they are only inhibited when you have to make a eye movement. And the omnipause neurons are situated in the dorsal raphe nucleus. And the dorsal raphe nucleus projects to the PPRF and the interstitial nucleus of the medial longitudinal fasciculus, which helps in the horizontal and the vertical movements. And this substantia nigra sends inhibitory output to the superior colliculus, right? And then the superior colliculus then is going to inhibit these two. Okay, so that's where the omnipause neurons are situated. So again, I would just like to show you. So sir just discussed, you've already had lectures on the substantia nigra, and I'd just like to tell you what is disinhibition. So you can see, so what I want you to know is that once you decide you have to make an eye movement, you will get a command from the parietal cortex to the frontal eye field, which I told you is the center for conjugate movements. The frontal eye field, now another thing I wanted to tell you was, none of the areas of the cerebral cortex project directly to the cranial nerve nuclei. Okay, none of the areas project directly to the cranial nerve nuclei. The frontal eye field projects to the paramedian pontine reticular formation. It also projects to the nucleus prepositus hypoglossi, which has a burst, and the tonic neurons, and it projects for vertical saccades to the rostral-interstitial nucleus of the medial longitudinal fasciculus and the interstitial nucleus of Cajal. But what I would like to tell you was, so the frontal eye field projects directly as well as through the superior colliculus. Okay, so the superior colliculus is under direct control of the frontal eye field, but it is also under indirect control of the frontal eye field through the caudate nucleus and the substantia nigra, the pars reticulata part, which sends out powerful GABAergic inhibitory output to the superior colliculus. Okay, so what happens? You could have direct stimulation of the superior colliculus by the frontal eye field, or when a movement has to be initiated, it activates the caudate nucleus. Okay, the caudate nucleus, once stimulated, sends out inhibitory output to the substantia nigra, and since the substantia nigra sends inhibitory output to the superior colliculus, by inhibiting the substantia nigra, it prevents the inhibition of the superior colliculus. This is known as disinhibition. So by activating the caudate nucleus, it inhibits the substantia nigra, okay, because this is the inhibitory output to the substantia nigra, and thus the inhibition of the superior colliculus is prevented. So it releases the superior colliculus, the inhibition on the superior colliculus, and then the superior colliculus sends fibers. These are the tectobulbar fibers. The tectobulbar fibers are then going to project to these reticular formation, which is there in the midbrain or in the pons, okay, and this is going to result in these movements. Okay, so the fibers cross and they go to that of the opposite side. Now this is a diagram from Barr. This is important for you, you should know, and I'll just explain that in detail. Now when you have, this is the pathway for horizontal saccades. Now when you have to do a horizontal saccade, as I told you, the frontal eye field gets information from, let me see, there's a, okay, for horizontal saccade, you get information from the, for visual attention from area 7. It activates the neurons in the frontal eye field. This activates, you can see fibers going directly to the paramedian pontine reticular formation, to the nucleus prepositus hypoglossi. Here you have the burst neurons, here you have the tonic neurons, and here both of these are then projecting to the 6th nerve nucleus. Okay, you can also see that the left frontal eye field projects to the superior colliculus. Again, fibers cross to the opposite side, go to the burst and the tonic neurons in these two groups of nuclei. Again, I told you in the 6th nerve nucleus you have two types of neurons. The neurons which are general somatic efferent, which are going to innervate the muscle of one side, the lateral rectus of that side, and the internuclear neurons, which are going to travel in this myelinated midline tract, the medial longitudinal fasciculus, and then it is going to supply the medial rectus of the opposite side, so that you can have conjugate movement of the eye. Again, I am repeating, the burst neurons which are going to initiate the movement are in the PPRF. The tonic neurons which are going to actively contract to maintain fixation are in the nucleus prepositus hypoglossi. The fibers are being relayed to these. No fibers of the cerebral cortex is projecting directly to the cranial nerve nuclei. It is being projected via the PPRF and the nucleus prepositus hypoglossi, right? and you can see that these neurons then terminate here in the in the sixth nerve nucleus, and the fibers cross the internuclear fibers which are traveling in the MLF to stimulate the medial rectus of the opposite side. Now just a little more in detail, just to explain to you that this is basically for the postgraduates. You can see that when you have, okay, this is the here are the omnipause neurons. The omnipause neurons have to be inhibited the time the saccade is generated, and these are situated in the dorsal raphe nucleus. Then you have the nucleus here, which is the abducens nucleus, and this is the paramedian pontine reticular formation. Now what's important is that when the lateral rectus of one side, I told you there are the burst neurons here, they stimulate the neurons of the abducens nucleus, but they send the inhibitory burst neurons to the opposite side, so that the lateral rectus of the opposite side is inhibited. You are also getting input from the medial vestibular nuclei, okay, and you have the the nucleus prepositus hypoglossi, which is for the tone, where the tonic neurons are situated. So for the vertical saccades, what's important is, you know that for vertical movements you have the superior and the inferior rectus, as well as the superior oblique and the inferior oblique. As I told you, the superior rectus is supplied by crossed fibers of the oculomotor nerve nucleus, and the trochlea, the superior oblique is supplied by the trochlear nerve, and you all know that fibers decussate. The fibers of the oculomotor nerve do not decussate, except the fibers which are going to supply that of the superior rectus. So again you can see here that you have the fibers from the cerebral cortex, the frontal eye field, go to the superior colliculus. These relay in the these fibers are going to relay in these two nuclei. This is the one which has the tonic neurons, this is the one which has the burst neurons, okay. From here the fibers are going to relay in the oculomotor nucleus, which is going to supply this muscle, the superior rectus of the opposite side, and the trochlear nerve of the opposite side, and passes through the posterior commissure to supply these other muscles of the same side, okay, because these fibers are always crossed, okay, they supply from the opposite side, okay. So this is for the vertical saccades. So then you have the smooth pursuit movements. The smooth pursuit movements are movements which are the movements which are done when you have to follow an object across moving across the visual field. And for this movement, the only additional center involved is that you have to have information from the middle temporal and the medial superior temporal areas, and I told you that is the junctional area between the visual cortex, the occipital cortex, the parietal cortex, and the temporal cortex. This calculates where the object is, what is the position and the velocity of the object, okay. So once the fibers have relayed to the visual cortex, the information is then it is going to calculate the velocity, the position of the object. Then the information is sent to the frontal eye field, and then also for calculating the velocity, the position of the object, you require information from the cerebellum, from the flocculonodular lobe, and the inferior vermis, from the vestibular nuclei, and then the pathway for the others is the same, that is the paramedian pontine reticular formation, to execute the horizontal, vertical, or the oblique movements, okay. And then you know for Vergence, you know that for Vergence the movements, like you saw, you've already done the optic pathway and the pathway for accommodation. The pathway, for example, you have the optic pathway, the fibers, for example, let me trace the pathway for Vergence. Convergence, for example, you have the optic tract, the lateral geniculate nucleus, the fibers will go to the primary visual cortex, okay. From here the fibers will then be projecting to the to the superior colliculus, to the corticotectal fibers, and then the fibers they go to the to the third nerve nucleus, the oculomotor nerve nucleus, to the medial rectus to cause Vergence movement. In addition, usually when you are looking at a near object, there is also increased curvature of the eyeball. So the fibers that are involved, then from the superior colliculus, the fibers go to the accessory, the Denga-Westphal nucleus, okay, through the through the nucleus of the optic tract, and then these fibers are going to from these From the olivary pretectal, from the nucleus of the optic tract, the fibers will go to the Edinger-Westphal nucleus, ciliary ganglion, and then through the short ciliary nerve supply the ciliary muscle as well as the sphincter pupillae. So that results in what is known the convergence, constriction, as well as the contraction of the ciliary muscle is going to increase the curvature of lens during accommodation. Okay, so smooth pursuit movements are nothing but movements in which an object moves across the visual field and requires more information about the position and the velocity from the parieto-occipital cortex. Okay, and there is input from the cerebellum and the vestibular nuclei. So this is showing you the same thing. Once the striate cortex to calculate where the object is by the middle temporal or the middle superior temporal areas, which are the junctional area between the occipital, parietal, and the temporal cortices. Then there is involvement and from the cerebellum, vestibular nuclei to exactly calculate the velocity of the object and how fast your eyeball should move, and then the pathways are similar. Okay, so we've seen what is a saccade, so what is a conjugate movement and a disjunctive movement. Conjugate movements are when the eyes are moving in the same direction, same amount simultaneously. These could be saccades, smooth pursuit movements, or fixation. Saccades are rapid jerky movements. These are voluntary movements which can be made in response to visual stimuli, acoustic stimuli, tactile stimuli, right? These movements, the center is in the frontal eye field. The burst neurons are situated for horizontal saccades in the paramedian pontine reticular formation. Tonic neurons for these pause points are situated in the in the nucleus prepositus hypoglossi. For vertical saccades, burst neurons are in the interstitial nucleus of the medial longitudinal fasciculus, and for the tonic neurons are in the interstitial nucleus of Cajal. Oblique movement saccades require the activation of both these pathways. Smooth pursuit movements are nothing but tracking an object moving across the visual field. They are slower movements. These are involuntary movements, and these movements require the involvement of the junctional area between the occipital, parietal, and the temporal cortices, which are named as the middle temporal and the medial superior temporal area in monkeys, and that is the one which actually calculates the position and the velocity of the object. And the rest of the path, it also requires information from the cerebellum and the vestibular nuclei to calculate exact velocity of the object, as well as to then calculate how fast your eye should move. Okay, then you have these movements which are optokinetic, which are sustained movements of the eyeball and the head. So the eyeball and the head move in the same direction. You have the vestibulo-ocular movements, which are brief movements, and since they are brief, rapid movements, so the eyeball, having lower inertia, moves first, and then the head begins to move. And once the head reaches its final position, the eye rolls back to lie in the center of its new position. Okay, then you have the disjunctive movements. The trigger for disjunctive movements or Vergence movements is blurring. That when you look from a near to a distant object or a distant to a near objects, the objects which are, for example, you want to focus on a near objects, the distant objects are blurred because of retinal disparity. And these Vergence movements involve eyes moving in different directions through different amounts. That's all. Thank you. Stay safe, stay calm, and stay blessed. Bye-bye. Done. ऐसे ही करना है एंड स्ट्रीम कितने थे हाउ डू यू नो हाउ मेनी अटेंडेड ओके नॉट एंड